Hello, my name is Kelmer Martins-Cunha. I'm an undergraduate student at Universidade Federal de Santa Catarina. I'm a member of Mycolab, Laboratory of Mycology from Universidade Federal de Santa Catarina, and I'm also a member of the MindFunga initiative, which stands for Monitoring and Inventorying the Neotropical Diversity of Fungi. And in this video I will present my ongoing undergraduate research project that is titled in this case Is Rigidoporus ulmarius a species complex? Some preliminary results. So Rigidoporus ulmarius is a polypore species that as a facultative parasite causes white rot in several tree species. It has a cosmopolitan distribution, and as we can see in this map, most of its occurrences are concentrated in America and Europe. So this species is morphologically characterized by a large basidiomata with 30 up to 150 centimeters long. This picture is actually from the biggest Rigidoporus ulmarius ever found. It has a pale buff to cream, sometimes pinkish buff to light buff upper surface, and in some specimens there is also a greenish tint that we can barely see in this picture. It has a monomytic ecosystem, globose to subglobose basidiospores, and the presence of fusoid cystidioles. So these morphology traits separates it from other Rigidoporus species, and this description can be found on the classical literature, if we want to call it so. That in this case is Neotropical Polyporus by Leif Heivaden. Recently, some specimens of Rigidoporus ulmarius were found in Parque Nacional de São Joaquim, Santa Catarina, Brazil, and in Nova Trento, Santa Catarina, Brazil, around the Monte Barão area. We were able to identify the hosts of the Parque Nacional de São Joaquim specimens that was occurring in a living Ocotea puschella. These are some photos of Ocotea puschella, and unfortunately for the Nova Trento specimen we are not able to identify the host in a species level because it was occurring in a dead trunk, but it was a dead trunk of an Ocotea species as well. And some morphological analyses were conducted for some of these specimens. And in a preliminary analysis, there are some morphological evidences between these specimens and materials from other areas, especially in basidioma color and basidiospore size. So these specimens displays a similar basidiospore size when compared to East African specimens, described by Heivaden and Johansen in 1980. But when they are compared to North American specimens described by Gilbertson and Heivaden in 1987, they show a smaller basidiospore size. There is also a disparity in the broad range of hosts for Rigidoporus ulmarius, with about 12 hosts identified in a species level worldwide. And as the Parque Nacional de São Joaquim and Nova Trento localities are considered high altitude environments, it seems that Rigidoporus ulmarius has a pattern association with Ocotea species in these particular environments. There are 3 known hosts for Brazil and South America: Araucaria angustifolia, which is also a high altitude species; our find, Ocotea puschella, and then occurrence from Argentina in Ocotea cutifolia. So as next steps for this research, This preliminary analysis show us that there are some evidences of a species complex in these specimens. So these morphological divergences, together with these ecological divergences and the cosmopolitan distribution of Rigidoporus ulmarius, suggests that the species could actually hold a species complex. And there's also the pattern association with Eukaryote in high-altitude environments. So these specimens could represent a new species lying inside Rigidoporus ulmarius, and that is limited to these high-altitude environments. So to test these hypotheses and corroborate our ideas, we will continue to conduct morphological analysis of different Rigidoporus ulmarius specimens, together with phylogenetic analysis and ecological and geographical comparisons as well. So if this hypothesis is confirmed and these high-altitude specimens really represent a new species, it could already be threatened due to its limited distribution in these high-altitude environments. So as examples, there's Androgena tropica and Fungal nubicula, two recently described high-altitude species that are already threatened. Both of them are considered vulnerable based on the IUCN assessment criteria. So I would like to thank you for your attention and for all the support and funding from these institutes and organizations. Thank you.